I'm just giving the intro about the marine biofouling aspect. If possible, I can go ahead with the mitigation measurements, what we are doing at our Center for Ocean Research. So today I'm going to talk about basically about the marine biofouling. We all know that some kind of marine organism, especially when we are walking in the beaches, especially in the rocky shores, we can see such kind of plants and animals sticking onto the rocky environment. If these flora and fauna sticking onto the rocky environment, it may be very ornamental, and it is very good when we are walking in beaches. But it will also occupy the man-made structure, and it becoming a pest to such kind of industrial applications. So these animals are called as biofouling, and there may be two kind of biofouling that may be soft fouling as well as hard fouling. The soft fouling is comprises of soft coral, sponges, anemones, tunicates, and hydrides, whereas the hard fouling is mainly based on barnacles, mussels, tube worms, bryozoans, and seaweed. And these are all some of the pictures to highlight about what is biofouling. You can see the ship hull. The base of the ship hull is completely fouled, and it is now taken for dry docking. And you can see a renewable energy platform that is completely fouled with the barnacles. And you can see a turbine which is used for wave energy generation. The turbine is completely occupied by the kelp seaweed biofouling. And also you can see a data buy which is completely fouled. If these data buys are fouled, even the sensor get fouled and the weight will be increasing. And you can see the bridges, even concrete structures are being fouled. And finally, the underwater structures also being fouled. Biofouling is not only going to be an addition or an attachment to a materials which is being made, like fishes, ship hulls, or any other concrete structures in marine environment, but is also indirectly providing some kind of negative impact for our climate change. They are triggering greenhouse gases because when there is a biofouling in the ship hull, the weight of the ship will increase. If the weight of the ship is increased, the normal sailing cannot be possible in the ship. So any navigator have to add more fuel to increase the speed or the knot of the ship. So when they are adding more fuel, there will be more emission of carbon dioxide that will lead to greenhouse gases. So that is also directly having an impact on climate change. And these are all examples where the power plant industry is being affected because of this marine biofouling problem. Because those power plant industry, especially like Kalpakkam goes where our Madras Atomic Power Station is there, and they are utilizing sea water as a secondary coolant agent. If the sea water is taken into the cooling water circuit, they are having the tendency to develop this biofouling. You can see the tubular heat exchanger as well as plate heat exchanger being fouled with green mussels and other biological entities. And all these oil rigs are being the main focus in especially in coastal and marine environment, and these platforms are also being fouled with such kind of organisms. And cage aquaculture, the net which is being used to cage the fishes or animals being cultured here or also have the tendency to foul. If the net is being fouled, there won't be any kind of a normal water exchange happening in the cages. So there may be some kind of less production will happen due to this biofouling, and that have to be eradicated. And these are all the other thing. You can see a sensor is completely fouled with barnacle, and you can see a data buy which is supported with the solar system also completely fouled with barnacle. And in the yellow color wave gauge, the tide gauge is completely fouled with barnacle. And the lower film, you can see a current meter, which is before and after deployment. When it is deployed, you can see the current meter, and after deployment, it is completely occupied by the goose barnacles. So there is a relation. It's a cycling process. We can say that any material which is being dumped in the seawater, or any material which is having a connection with an aquatic media, the interface, liquid and solid interface, have the tendency of biofouling, and that biofouling have to be cleaned. That is called as anti-fouling. So this marine biofouling can be classified as microfouling as well as macrofouling, and this microfouling and macrofouling will happen in a sequential manner, which is tabulated here. You can see the process. All the rose color boxes are representing the microfouling, and the green color boxes are representing the macrofouling. The microfouling starts with the initial deposition of organic molecule. See, this is nothing but the biomolecules such as carbohydrates, proteins, lipids, and other any kind of secondary and primary biomolecules available in seawater. They will come and stick onto the material surface. It may be a ship hull, or it may be a boat surface, or it may be a cage net, or it may be any kind of a underwater tube, or any sensors, or any kind of a concrete platform. Immediately once the water and the surface interface is having the link, within a second this deposition will happen. This is called as initial deposition of organic molecules. And this organic molecule film will be attracted for the bacteria which is present in the aquatic environment, especially in the marine environment. These bacteria have a tendency to attach to the surface. Some may have the firm attachment, and some may not have the firm attachment. So the adhesion may be reversible and irreversible. Those are reversible, there won't be any problem. There won't be any problem in the biofilm formation. But if it is irreversible, there will be a biofilm formation. Once the bacteria started attaching to a surface in an irreversible manner, they develop the quorum sensing behavior, and through the van der Waals force, as well as with the slime formation, they started doing the slime formation activity and EPS secretion. So the entire biofilm will be very sticky, and that will attract the diatom and algae. So the biofilm will be enriched with diatom and algae later in the marine biofilm in the later stage. So the biofilm is very thick with organic molecule, bacteria, EPS, especially polysaccharides, as well as with diatom and algae. When it is rich with such kind of a microfouling, that will be an attractant to the larvae as well as to the spores. Okay, these microfouling, they will give a chemical clue to those larval as well as spores, and they will be attracted by those chemical clues, and they come and settle onto this. Once this larval settlement happen, that will be that is called as succession of larval recruitment, and that will be developed as a biggest macrofouling, and that is called as macrofouling. Okay. So this is taken. The image is taken from a PLOS One journal, especially with the author Martin, who published this paper in 2015. He is just giving a glimpse that the biofilm starts within a minute. I can even say that it will start within a second. And the particle of organic matter will come and settle, then the primary colonizer, then secondary colonizer, tertiary colonizer, and become a macroscopic fouling community. And the microfouling, they are the heroes like bacteria, diatom, and the macrofouling will be taken back here by barnacle, green mussel, brown mussel, as well as tube worms. And these bacteria cannot be seen in the naked eye, so we have to work on any kind of stains, or we have to do any kind of lab-based culture. and we have to seek and we should know what is a bacteria. And diatom, we can see through a microscope. You can see a beautiful structure. They are all having the tendency to stick on the fouling surfaces. And you can see these are all the, you can see a camera in the middle of this slide, which is retrieved from the ocean. You can see how barnacle are being attached to on the camera surface. And these are all the barnacles. Even you can, I can say that the Predator movie is being mimicking approach of this barnacle only. So these barnacles, they are mainly having a great shell on the outer surface, and they are arthropod, especially coming in the insect family. And they have a sessile stage where they are firmly attached like this, and they are also having a free swimming stage. Once the adult started uh, doing the reproduction and releasing the fertilized egg in the open environment, it is in a free swimming stage, and the metamorphosis happen, and they will end up with the cyprid stage. Cyprid stage is the stage where they have the pseudopodia with a walking leg that will start walking on the surface, where microfouling is interstressed with. There they will come and settle, and they will grow as an adult. So this will be the pattern for all the barnacle, and that once the cyprid is being settled in a surface, they secrete the cement protein, and there the biofouling starts. This is the same case for green mussel also. They are also having a free swimming trichopod, develinger, pedivelinger, as well as pad settlement. These pad are the metamorphosis stage where they search for a surface, and they come and settle on the surface and as develop as a adult green mussel. And adult green mussel once settle on a surface, it is not easy to remove those mussel fouling. Apart from that, even seaweed spores also coming and settling to surfaces. You can see a propeller is completely meshed with the seaweed fouling thing, as well as the tide cage is completely fouled with the seaweed spores, as well as barnacle larval settlement. And I already said that marine biofouling is the biggest menace, and government and industries are almost spending 5.7 billion US dollars annually to prevent this fouling process. This is called as anti-fouling, and it may be a physical cleaning, chemical treatment, mechanical cleaning, and anti-fouling paint-based cleaning. And also nowadays people are also the tributyl tin-based as well as copper-based anti-fouling paints, which is already used before 2003 was being banned in the anti-fouling industry, and now people are working on less toxic metal-based coatings, natural-based eco-friendly based coatings, as well as biodegradable polymer-based coatings, as well as nano-based coatings. And as I already mentioned, that a clean surface which is immersed into an seawater environment, it will lead to a microfouling. That microfouling will provide a slime formation that will attract the bacteria diatom as well as larvae, and that is called as macrofouling. And that macrofouling had to be cleaned. The cleaning process is called as anti-fouling. This is always happening in the ship industries and other industry where seawater is being used. And I'm just being due to the time constraint, I'm just ending up this slide. And these are all the facilities available at the Center for Ocean Research to do any kind of marine biofouling related research. So we have developed a marine bacterial fouling test, we will do biofilm thickness, biochemical composition, biofilm chlorophyll, as well as ATP analysis, live dead analysis of biofilm, and next generation sequence based metagenomic analysis of microfouling we are doing. And apart from that, in marine diatom fouling, we are working on how the diatom is being adhered to the surface, and we have already developed a technology to count those diatom, and we are also developed technology for live data analysis of those diatom, as well as we are working on next generation based enumeration of marine diatom populations. Apart from that, we are also helping people to test macrofouling larvae. We are culturing green mussels as well as uh, seaweed. Here we can work on green mussel spore settlement, so green larval settlements as well as seaweed spore settlement. And we are also helping people to do any kind of field exposure studies in Muntam Harbor. We can completely helping in to test the biofouling aggregation in any kind of testing surface, seasonal studies as well as larval recruitment on, and also we are helping the to know what kind of biomass is accumulating on the surface and what is the weight increment in the surface. And we are also helping people in testing the cleaning efficiency. Slides also what we have done in our laboratory, especially on control measurement. We have done some kind of an experiment on nano-based control, marine natural product based control. We have also developed polymer based coatings, and we have also developed surface engineer based coating also. If time permits, I will do that kind of information. I will present those information in next series whenever time permits. Thank you for everyone for listening in my topic in a very patient way. Soumya? Soumya.
Yes, sir. Yeah. Thank you so